want to provide you with a stripped back basic explanation of how ultrasound is generated and detected by our probe surface. Sound is simply the transfer of mechanical energy from a vibrating source through a medium. Ultrasound is a high-pitched frequency of sound which is above the audible human range. Typically, ultrasound probes generate sound at a range of around 4 MHz to 15 MHz in frequency. This is considerably higher in frequency than the audible range of hearing. A vibrating source is necessary to create that initial pressure wave. So if we think about the inside of a bell, when we ring the bell, the clangor of the bell hits the side of the bell and it creates a vibration, and this is how the sound is transmitted onwards. An ultrasound probe is really called a transducer because it converts one form of energy into another, i.e. it converts electrical energy into sound energy. The ultrasound probe is designed to produce steerable beams of sound. So each individual ultrasound beam is composed of multiple scan lines that are produced in sequence across the surface of the probe. Each of these scan lines can then be represented as lines on our ultrasound image in the form of pixels. So how is ultrasound generated? We require a unique piezoelectric crystal in order to generate sound, and these are found at the surface of the transducer. They're arranged in lines in groups across the whole of the surface front. The piezoelectric crystal is unique in that if we apply an alternating voltage to that crystal, the crystal will subsequently expand and contract as the alternating voltage occurs. Each group of crystals generates a single scan line, and these scan lines are produced in sequence. If we think about our orientation marker on our probe, this dictates the crystals which are first fired in any sequence of our ultrasound image. Hence the importance for correct image orientation. The frequency of the voltage applied determines the rate at which the crystals vibrate and contract. This subsequently has a knock-on effect on the frequency of the transmitted sound. So if our crystals are vibrating at a faster rate, we will have a higher frequency beam generated. So in essence, to generate an ultrasound beam, we require piezoelectric crystals. These crystals need an alternating voltage to be applied to them to cause their surfaces to expand and contract. The rate at which these crystal surfaces expand and contract determines the rate at which our ultrasound frequency is transmitted. So in order to detect oncoming ultrasound or a returning signal from the body interfaces, the sound reaches the probe crystals and applies a force to those crystals. The strength of the returning signals is proportional to the force applied to the crystals by those sound waves that come back from the reflected surfaces. The magnitude of this force in turn generates an alternating current. So the strength of the signal returning equates to the strength of the force applied to those signals. So if we have a signal returning from bone, which is a highly reflective surface and a strong attenuator, then we will have a strong force applied to those crystals, and the signal created by that alternating voltage will be a high signal or a bright signal on our screen. Conversely, any returning signals from the body which are weaker, for example, those returning from fluid within the bladder, will have a very weak force applied to the crystals. This creates a low signal, and therefore we have a dark or anechoic signal on our screen. The amplitude of the signal returning to the probe will determine the brightness of the image we see on our screen. This is how we can differentiate between tissue types on our ultrasound image. So we have at one extreme, we have the mineralized substances such as calcification or bone. They will be highly reflective and they will appear as bright signals on our screen. The other end of that spectrum, we have our poorly reflective structures such as anechoic fluid or blood within the vessels. They will be displayed as dark or anechoic structures on our ultrasound image. In between these two will be a whole range of soft tissue structures, and they will be displayed as a range of grey shades on our image. So in conclusion, we need an alternating voltage to apply to our crystal surfaces. This generates a vibrating force and consequently an ultrasound beam. When our signals return from the body, they apply a force to our crystal surfaces, which is transmitted into the form of a signal. That signal is displayed on our ultrasound image as varying shades of black or white and brightness, and that determines the type of tissue that we have been scanning.